All right, okay. Hello, and I'm pleased to welcome all of you to the ZOOMinar. We have two presentations today focused on AFM-based studies on protein aggregation, and the speakers are Dr. Kenjiro Ono from Kanazawa University, Japan, and Dr. Justin Legleiter from West Virginia University. It's my pleasure to introduce the first speaker today, Dr. Kenjiro Ono. Dr. Ono received an MD degree from Showa University, Tokyo in 1997, and a PhD degree from Kanazawa University in 2002. He did his postdoctoral research with Dr. Teplo in UCLA from 2007 to 2009. He then went back to Japan to join the faculty of neurology at Kanazawa University. In 2016, he moved to Showa University to become the chair and full professor of neurology. But then he returned back in 2021 to become the chair and professor of neurology in Kanazawa University. So he's at Kanazawa University, and he has received many honors and awards, including the 2015 Japanese Society of Neurology Award and the 2017 Kazoto Prize from the Kazoto Research Foundation. With that brief introduction, Kenjiro-san, thank you very much for joining us. I know it's very early morning to you, so ohayo gozaimasu, don't fall asleep, get started.
Thank you so much. Hello, everyone. Could you hear me?
Yes.
Thank you. My name is Kenjiro Ono, Department of Neurology, Kanazawa University, Japan. Before my presentation, I will express my gratitude to Professor Ramamushi and organizing members. Today, what I would like to talk about is high molecular weight Aβ oligomers are important targets of disease-modifying approach of Alzheimer's disease. COI disclosure is here. Okay, at first, I will mention the direction of research and therapy of Alzheimer's disease. There are two approaches. First is the approach from neurotransmitters. Second is the approach from pathology and biochemistry, that is protein aggregation. First approach, for example, cholinesterase inhibitor, such as Dmt, may be limited to symptomatic therapy. And so, based on the recent findings of basic and clinical researches, now we are focusing on second approach. That is disease-modifying therapy, DMT. This slide shows neuropathological findings of AD. Upper panel are macro findings, and lower one, micro findings. As you see, AD brain shows parieto-temporal atrophy, including hippocampus. Upper panel in micrograph. There are three characteristics. As you know, senile plaques, composed of Aβ, neuroferro tangle, composed of tau, and neural death and neurodegeneration. This slide shows Aβ cascades. This hypothesis proposes a series of pathologic events leading to AD. Cerebral Aβ protein accumulation is the primary factor in AD, as you know, and the rest of the disease process results from an imbalance between Aβ production, accumulation, and Aβ clearance. Based on Aβ cascades, there are three targets of DMT. First is beta or gamma secretase inhibitor. That is inhibition of Aβ production. Second is Aβ degradation enhancers. For example, neprilysin and insulin degrading enzyme. And final is inhibitor of Aβ aggregation or removal of Aβ aggregates with immunotherapy. Although we previously had focused on final aggregate of Aβ mature fiber, but based on the result of clinical trial of DMTs and clinical study, such as DIAN study, the targets have been shifted to early or intermediate aggregates, as you know, such as soluble oligomer and protofiber. That is oligomer hypothesis. In 1997, using site exclusion chromatography mainly, my boss Dave Teplov firstly discovered a novel intermediate in the pathway of Aβ fibril formation. He called the Protofiber for these aggregates. After that, his group reported that Arctic APP mutation E693G enhances protofiber formation without influencing fibril formation of Aβ using Arctic model. In 2007, ELISA using anti-protofiber antibody was invented for detection of protofiber in biological samples. And then this anti-protofibril antibody was confirmed to be safe and tolerable in the phase 1 study and proceeded to the phase 2 study in clinical trial. In 2018, at AAIC Chicago, it was reported that phase 2 clinical trial using this antibody could reduce amyloid burden as well as significant suppression of cognitive decline. At now, phase 3 clinical trials are currently underway. This slide shows the observation of fibril formation from low molecular weight Aβ, mainly monomers, by using high-speed AFM, collaborated with Takahiro Nakayama, Kanazawa University. Let's see. At the beginning, it takes time. This is first phase, the nucleation phase. This phase taking time. After this critical nucleation phase, and then we could observe clear fiber extension of Aβ 142, which is second rapid phase, extension phase. And the next slide shows the observation of fibril formation from high molecular weight oligomer, protofiber, by using high-speed AFM. Let's see. Even after the time when the monomer start had already formed enough fiber. In the case of protofiber start, interestingly, only a few fibrils were formed. In addition, we directly observe the association with protofiber itself. Based on our result with high-speed AFM, protofiber may not always on pathway from monomer to mature fiber. Maybe protofiber may supply monomer from protofiber itself. This slide shows the result of cellular experiment. MTT, WST, and LDH assay. Red is monomer, blue protofiber. All assays show protofiber are more toxic than monomers. These figures show calcium etidine homodimer assay showing protofiber exerts more membrane damage. results of LDH and calcium HDM homodimer assay that reflect cell membrane damage were clearly different between monomer red and blue protofiber. But in the MTT and WST assay using iPS neuron that reflects mitochondrial enzyme activity, the difference was not big. From these results, in short-term Aβ 142 treatment, Aβ may first attack the cell membrane and subsequently induce damage to mitochondria. The degree of cell membrane damage was more severe in Protofiber than low molecular weight Aβ monomers. Other group, here Rushwell, also reported that Protofiber exert neurotoxicity via membrane damage, such as carpeting, pore formation, detergent effect, and so on. Left picture are the result of AFM. These images show pore formation on mimicking membrane. This slide shows that loss degeneration significantly increased in cell-exposed either low molecular weight or protofiber Aβ for 30 minutes or untreated control cell. Consistent with above described cell toxicity assays, high molecular weight Aβ protofiber resulted in greater oxidative stress, as evidenced by more intense green fluorescence than cells treated with an equivalent dose of LMW Aβ. Next slide shows membrane lipid peroxidation is enhanced protofiber Aβ 142. Lipid peroxidation was measured by DPPP assay in cell after treatment with low molecular weight red or blue protofiber. Again, both Aβ 142 fraction induced significant membrane phosphorate peroxidation compared with white box untreated control cell, but the severity of peroxidation as estimated by fluorescence intensity was greater following protofiber treatment compared with low molecular weight Aβ. This slide shows protofiber as well as low molecular weight Aβ significantly reduced cell membrane fluidity compared with non-treated cell. Furthermore, Protofiber produced a larger decrease in cellular fluidity than did low molecular weight Aβ. Consistent with the result of the cell membrane fluidity assay, Protofiber led to a decrease in membrane fluidity as evidenced by a darker blood level of ratio image than untreated cell. Individual ratio images of cell untreated, treated with low molecular weight Aβ or protofiber are shown in right three lower panels. We next investigate the effects of low molecular weight and protofiber Aβ 142 on calcium ion homeostasis. Please see left panel. The peak increase did not differ significantly between low molecular weight and protofiber Aβ treatment, but the plateau level was higher in cell exposed to protofiber after 60 seconds of exposure. Please see right panel. Pretreatment with nicardipine caused significantly in this calcium ion elevation measured after 75 seconds of protofiber Aβ exposure. These findings suggest that protofiber promotes calcium influx through L-type VDCC in cell. Furthermore, in exposure to low molecular weight or protofiber pretreatment with nicardipine plus memantine resulted in a marked reduction of calcium ion compared to pretreatment with nicardipine alone. As you know, memantine modulates the glutamatergic system, likely by blocking calcium influx through NMDA receptors. This slide shows the result of neurophysiological experiment. Red circle is low molecular weight A beta, blue protofiber. Finally, we demonstrated that protofiber significantly inhibited long-term potentiation synaptic plasticity in the CA1 subfield of hippocampal slices of mouse. Our result suggested that loss generation and decreased cell membrane fluidity induced by protofiber A beta impair the molecular, cellular, and synaptic properties essential for learning and memory. Furthermore, membrane formation may also impair cellular and synaptic function, and it is consistent with the observed loss of membrane integrity. Taken together, our results demonstrated multiple mechanisms of A beta protofiber induced neural injury and death. We have previously used high-speed AFM to prove reversible inhibition of A beta aggregation by miricitine. Please see left panel. With A beta 142 monomer and miricitine, even after 60 minutes, only a small amount of A beta aggregation can be found, even after 1 hour. On the other hand, please see right panel. Once miricitine was removed here, A beta aggregation was restored surprisingly. And so we have proved reversible inhibition of A beta 142 aggregation by miricitine. Last year, we reported that miricitine prevents protofiber-induced neurotoxicity. Through antioxidants in cell membrane. This slide shows the result of loss production. Please see blue box. Loss production increased in cell treated with protofiber compared with control cell, gray box. Please see orange box. Miricitine treatment significantly reduced loss induced by protofiber. In fluorescence images, right panel, the green fluorescence was enhanced by protofiber, but it was dimmed by miricitine treatment. This slide shows the result of lipid peroxidation in cell membrane, DPPP. Please see blue box. Cell treated with protofiber showed a significant increase in lipid peroxidation, which was partially but significantly blocked by orange box with miricitine. Please see light box panel. DPP staining results are shown in light panel. After exposing cell to protofiber itself, increased blue fluorescence was observed. Whereas miricitine treatment reduced the fluorescence significantly. Next slide shows the effect of protofiber and miricitine on intracellular ionized calcium concentration of cell. Blue circle, high molecular weight Aβ oligomer protofiber significantly increased cell calcium ion concentration immediately after treatment, and whose increase was significantly reduced by orange circle with miricitine. Miricitine also reduced neurotoxicity through mitochondrial antioxidant activity. As shown in left panel, see blue box. Mitochondrial loss level increased in cell treated with high molecular weight Aβ oligomers protofiber compared to gray box control cell. However, orange box, miricitine treatment significantly reduced, but the protofiber induced this increase. As shown in light panel, cell treated with protofiber itself showed an increase in red fluorescence intensity compared to control cell, confirming an increase in mitochondrial loss. On the other hand, cell treated with protofiber and miricitine reduced this fluorescence intensity compared to cell with protofiber. Broad et al. Reported that Aβ protofiber level correlates with special learning in Arctic AD transgenic mice. Left panel shows that in younger 2-month-old mice, Aβ protofiber level was much higher than other mice. Right 2 panel shows that learning was modestly impaired in TG Arctic Swedish mice compared to non-TG litamate, with a significant effect of both genotype and time. This slide shows Aβ level in TG Arctic Swedish mice without plaque pathology and their relation to special learning. Left panel shows that Aβ protofiber level were inversely correlated with the improvement in escape latency, measured as performance in the last trial, subtracted from performance at the first acquisition session. Please see right panel. This panel shows that the level of total Aβ in the same set of mice were not associated with improved escape latency and special learning. Next slide shows that Aβ protofiber selective antibody prevents amyloid formation in a mouse model of AD. Upper panel shows Aβ burden in the cerebral cortex assessed with 6E10 immunostaining was much lower in younger TG Arctic Swedish mice treated with the protofiber antibody as compared to mice treated with control IgG. In lower panel, in elder mouse, 13 to 14 months, or TG Arctic Swedish mice with anti-protofiber antibody had approximately 75% lower level Aβ protofiber. On the other hand, there was no significant difference in immunohistochemical Aβ burden between both groups. Okay, I want to mention conclusion. First, high molecular weight Aβ oligomers protofiber, which are in the intermediate aggregates, can be important disease-modifying targets of Alzheimer's disease. Second, protofiber may not always represent the on pathway of Aβ142 aggregation. Third, protofiber may exert cytotoxicity mainly through membrane damage. Finally, miricitine polyphenol prevents high molecular weight Aβ oligomer protofiber-induced neurotoxicity through antioxidant effect in mainly cell membrane. Finally, I want to show acknowledgement slide. I am very sincerely grateful Canada University Neurology members, Professor Naiki, Fukui University, Professor Nishijo, Toyama University, and Professor Kiuchi and Tsuji and Oguchi, Showa University members, Department of Neurology, Showa University members, and AFM collaborated with Takahiro Nakayama, Canada University. And finally, I am so grateful for Michael Zagorski, Case Western Reserve University, and my two precious boss, Giulio Pacinetti, Mount Sinai, New York, USA, and Dave Tepro, UCLA, USA. Thank you so much for your attention.
Thank you very much for the very nice presentation, Kenjiro-san. Let me get started the questions for your talk. How big are these protofibers and how big are the pores formed by protofibers in the membrane?
Yes, I use same protocol in Dominic and Tepro protocol in mainly used side exochromatography. And after collected with protofiber peak, firstly, we confirmed morphological characteristics in Assay with first report. And then we confirmed the size of protofiber. And then we used other characteristic experiment. And next question, cell membrane damage?
Yeah,
Yes.
how big are the pores that you observe through AFM on the cell membrane or lipid bilayer?
Yes. Up to now, in an AFM experiment, we not yet perform AFM mimicking membrane and protofiber interaction. But from now, we have to the AFM observation. Yes, this is important.
So you mentioned that these protofibers can be on pathway and off pathway.
Yes.
Are they exactly the same? Are they different in their property and toxicity if they're off pathway or on pathway?
Yes. That's the important point. Thank you so much. We in cellular experiment, we use maybe off pathway protofiber we use. And yes, okay. And we confirmed protofiber if taking time, finally formed mature fiber. But compared with monomer start, it takes time.
Okay, can you look at the chat box? Let's go. There are many questions. Let's go from the top.
Thank you so much.
Chat folder. There's a question by Tom Rothstein. What is the strict definition for protofibers? How do you separate from oligomers and protofibers, oligomers and fibrils?
Yes, this is an important point. I think protofiber as in classical original paper, Protofiber may be about 20 miles, right? And but we sometimes when we submitted to Protofiber paper to journal, international journal, the reviewer pointed out most important point is morphological characteristics. And so we firstly compare to morphological characteristics.
Okay.
But hi, yes.
There's another question by Romani Abskaran. Great talk. How can you prepare the high molecular weight oligomers and how do you keep them stable?
Yes, this is also important point. Thank you so much. Yes, prepare, as I mentioned before, using side-section chromatography. But at first, we in order to get more efficiently get protofiber, firstly we incubated 2 or 3 hours, and then injection to side-section current. And yes, keep stable. This is also important for the point. Usually, we keep in minus 80 degree in the freezer. Yes, this is also important point.
So there's a question from Yuri Lyubchenko. What was the concentration of aggregates during the experiments?
Yes, this is also important point. In AFM experiment, we use 2.5 or 5 micromolar concentration. And in cellular experiment, also 5 micromolar concentration of A beta. This is also important point. Thank you so much.
There's a question from Sunil Kumar. Nice talk. I wonder about the mode of action of the polyphenol. I think it's polyphenol on the high molecular weight protofibrils of A beta. Could the polyphenol have the same impact on fibers or low molecular weight protofibrils? Has it been checked? I'm interested in the specificity of the polyphenol against the protofibrils.
Yes. Yes, this is also important point. Thank you so much. Yeah, when I studied in Tepro laboratory in UCLA in JBC paper in 2008, we reported that protofiber inhibited all stages, low molecular weight oligomerization, protofiber formation, and mature fiber formation. And so there is a possibility that polyphenol may be first initial stage of aggregation. And so we have to compare specific inhibition or not specific inhibition or initial inhibition of oligomerization. Yeah, this is also important for that. Yeah, we are deeper. We have to deeper work.
So I have a related question to that. This polyphenol that you used, what is it called? Miricitine, right?
Yes.
If you look at in one of your data, calcium ion flex, the rate of buildup of calcium ions is similar for the sample without and with miricitine. Can you go back to the slide and look at the...
Okay.
Okay, it's okay. You don't need. We can look at that at the end. So it looks like you do have reduction at the calcium level overall, but initially they rise very similarly. Go back several slides.
This is this slide?
Yeah, this is the one. Yes, yeah.
Yeah.
Look at the blue and the orange. Yeah.
Yes. You say this point?
Yeah, up to that point, they're very similar.
Yes. Yes, this is, yeah, I also wondering why. But even after 75 or 100 seconds, difference is clear. And so one possibility is that, yes, Yes, Miricitine, what stage, if it, yes, this is also important, but sorry, at now we don't have.
We can discuss it at the end. It looks like if it is inhibiting the protofibers or destabilizing them, it should happen in the beginning itself. I wonder why it didn't happen that way. Anyway, let me go to the next question.
Yes, yes, it's an important point. Thank you.
Let's take the question from Tom Rothstein. Do protofibers exist only briefly on the way to becoming fibrils? Are they stable as PF over time? I think they are stable, right? We're able to isolate them, able to keep them at minus 80 degrees Celsius.
Yes, 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 but yes, but I think yeah, it is better that we have to use this protofiber. Yes, yes, and finally, protofiber could change to mature fiber, but again, I want to mention take time.
Mm-hmm. Depending on the temperature, maybe the time it
Yes.
takes to convert.
Yes, yes.
So there's one question from Yukun Zhao. Can I know the information of AD mice? Where did you buy them from?
Again?
Can I know the information on the AD mice, Alzheimer's disease mice? Where do you buy them from?
Yes, yes, yes. We purchased in TG Arctic or mice from commercially purchased, and so in our group itself could not make TG mice.
So can you provide that information where you buy from in the chat box after your talk? Not now. I mean, you can do that later. Is it okay?
Yes, 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 yes. Thank you. Yes.
So let's go to the Q&A folder. There are a couple of questions. Let's go from the top. Are you ready? Okay. Yes. So the first question from Leif Anton-Schmidt. When you grow amyloid fibers on SLBs and investigated by high-speed AFM, do you see only single filament fibrils or also dimeric ones?
Again, single.
Do you see single filament only, or do you see dimeric fibrils?
Yes, yes. As yes, yes. At now, we could not observe the distinguish. Could not distinguish, but there are both possibility, I think.
Okay.
Yes, yes.
So next question is from Veloidam. Does miricitine bind to PF protofibers? Do they bind directly, or how do they work?
Yes, this is also important point. In 2012, in JBC paper, we confirmed miricitine could bind with Aβ monomer, but at now we not yet protofiber binding. This is
so
also
they bind
important.
to protofibers directly.
Yes,
right?
yes, 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 not directly.
So when they bind, do they destabilize, make them small oligomers or monomers? Do you have any idea?
Yes, yes. Using BIACOA SPR assay and NMR, we previously confirmed low molecular weight or Aβ monomer with bind directly.
And which are not toxic. Okay.
Yes.
So the last question is from Danilo Millardi. Phenolic compounds are somehow double-phased anti-amyloid, anti-AD compounds. They are both a source of electrons, which is antioxidant, but their aromatic rings may also interfere with phenylphenol stacking, which is believed to ignite amyloid toxic aggregation. Which of the two effects is preponderant in your results?
Yes, yes, yes. This is also point I estimated, but after now we not examine this point electron aspect sorry, but we have to this different mechanism. Thank you so much.
Thank you very much for your nice talk. Just stick around if we can. We'll have more discussion at the end of Justin's presentation. Thank you.
Thank you so much. Thank you so much.
Justin, you can share your screen and display your slides. Okay, our next speaker for today is Justin Legleiter from West Virginia University. He received his PhD in physical chemistry from Carnegie Mellon University. His thesis research focused on Aβ aggregation using by developing AFM techniques in the Kowaleski lab. Then he did his postdoctoral research for 3 years at the Gladstone Institute of Neurological Disease at UCSF, where he studied aggregates of Huntingtin fragments in the Machowski lab. In 2008, he joined the Faculty of Chemistry at West Virginia University. He was promoted recently to full professor. He has served as the associate chair of the department and as the director of graduate studies. He has been working on protein aggregation and developing AFM techniques. Justin, all yours.
All right, thank you for that kind introduction and for the invitation. This whole seminar series has been great, not only for me, but my students, and I appreciate. And I'm very excited to be able to present some of our research to everyone in attendance as well. So the last talk was actually really great, covered a lot of the introductory stuff that I would have gone over, but as a quick refresher, of course, we've got several neurodegenerative diseases that are associated with protein misfolding and aggregation. So for some reason, you get a protein that misfolds. It can be a mutation, some kind of processing event, but you're going to get these oligomers. And here we've got oligomers of different sizes, as a parent is going to be important. You can get your protofibrils, your annual aggregates, your amyloid fibrils, and all of those things are going to clump up together in tissues within the brain. And so for an example, here are some different depositions of beta amyloid or tau associated with Alzheimer's disease, depositions of alpha-synuclein for Parkinson's disease, and then a nuclear inclusion for the Huntingtin protein for Huntington's disease. Now, this aggregation process is really a complicated matter, right? And the environment in which those proteins are aggregating plays a huge influence on how the process plays out. And one of the things that I think is often overlooked is how important surfaces can be in controlling that aggregation process. So here, and one of these is not playing. Let me turn my pointer off. That worked just a minute ago. Well, you're not going to see it, but basically, if you have aggregation occurring on the surface, and here is wild type Aβ aggregating on the surface. So you see these high molecular weight oligomers, you see them moving around, and eventually they'll start to link up into many protofibrils. If you put a mutation in there, the aggregation looks completely different. And for some reason, what we checked was working a few minutes ago is not working now. But this looks completely different. Surfaces play a huge role in how these aggregates are actually going to form. And so that's what my talk is going to focus on. But when you think of a cell, the surfaces you're going to be interacting with are going to be membrane-based surfaces. So the plasma membrane, the surfaces of organelles. So if you look at this cell, there's all kinds of surface interfaces with cytosol, with extracellular space. And these membranes, the main component of these are going to be lipid components. So you're going to have all kinds of different lipid components. And so what can happen is as these proteins interact with the surface, that surface interaction can dictate the types of aggregates you get, potentially stabilize specific type of aggregates. But as we saw in the last talk, those membranes can also be targets of damage caused by these aggregates interacting with them. And so that's kind of where my lab has focused the last couple of years, looking at beta amyloid, but more recently, a lot more with the Huntingtin protein involved with Huntington's disease. So I'd like to give a quick introduction on Huntington's disease and some of the interesting features of it. So Huntingtin is the most common genetic neurodegenerative disease. It's caused by an expansion in the Huntingtin gene in which you have a CAG repeat. This CAG repeat encodes a polyglutamine stretch in the Huntingtin protein near the N-terminus. And so, turn my pointer back on. And so, if you look at, now my mouse isn't working. So now if you look at this protein near the N-terminus, you've got this polyglutamine stretch, if it gets past a certain length, typically around 35 glutamines in a row, this causes a disease. And the longer that glutamine stretch or expansion becomes, the more severe the phenotype is. So for example, the longer the stretch is, the earlier you have age of onset in Huntington's disease. Now, this polyglutamine stretch is also flanked by different sequences. So there's one called the N17 or NT17 for the first 17 N-terminal residues of Huntingtin exon 1, which precedes the glutamine. And then after that is a proline-rich domain. And these both influence the aggregation of Huntingtin. But what really drives it is this polyglutamine length. That polyglutamine length directly leads to aggregation. So if you have an expanded polyglutamine length, you can get all types of different aggregates. So here we've got oligomers of different sizes. We have annular structures and tiny pore structures here. Your classic fibrils, these amorphous clumps, and even these big clumps of fibrils that can come together. And you can observe all of these with a variety of techniques. These are AFM images here. Now, if we change that polyglutamine length in Huntingtin exon 1, so here we've got 20q, 35q, 46q, 53q, you can see as a function of time, the aggregation becomes much more aggressive. So you've got larger fibrils showing up earlier and taking over at earlier time points in the aggregation process. One thing to point out though, is even with a non-disease length polyglutamine stretch here at 20q, we still see oligomers. Over this time frame, we didn't see fibrils, but we do still see oligomers. And we're going to take that advantage of that later on down the road as well. But what we're really interested in is how this protein interacts with lipid membranes. And so there are a lot of different things that my lab has looked at over the years. I'm just going to review some of those before we get into some newer data. So first of all, the obvious first question is, what does polyglutamine length do? And so if we use our AFM in solution, we can make a supported lipid bilayer. So this is a total brain lipid extract bilayer forming here. Let it go long enough, you get a nice, smooth looking bilayer. What we do is we expose this to amyloid proteins and track what happens as a function of time. And so our first experiment here, we're just going to change the polyglutamine length. We're going to have Huntingtin exon 1 with 35, 46, or 51 glutamines in the protein. What we see when we add this protein is it directly binds to the membrane and starts to change the morphology. You start to see little tiny oligomers showing up, and it makes patches of the membrane much, much more rough. And we can quantify this roughness by measuring, the RMS surface roughness within those patches. We can also measure how much of the bilayer has those patches. And as you can see in these example AFM images, that as you increase the glutamine length, the aggressiveness of the protein in damaging that membrane or changing the membrane morphology is larger. It's polyglutamine dependent, which isn't too surprising because polyglutamine increases the propensity for aggregation. But what's also kind of interesting about this is if you measure the mechanical properties of these changed patches, you can see by monitoring, like for instance, the imaging forces, that there's a distinct mechanical difference between these regions that have altered morphology and the regions that still remain smooth. And so this is a histogram of the maximum force images, or maximum forces in this image for each individual pixel. And you can see the normal bilayers here, the maximum imaging force gets smaller, indicating that these regions are much softer than the other regions. So we know that it's damaging the membrane, or at least changing their mechanical properties as well. But the glutamine is only one part of the story. We've got the N17 region and the polyproline region. Ron Wetzel did some really beautiful work that showed, for example, that polyprolines tend to slow aggregation when attached to glutamines. And the N17 accelerates aggregation by forming an amphipathic alpha helix that assists in the formation of alpha helix rich oligomers that bring the glutamine stretches close together and accelerate aggregation. So my lab wanted to see if these flanking sequences impacted the interaction on membranes. And so we use these different peptides and basically just track the aggregation on a membrane surface again, total brain lipid extract bilayer. If we had just a pure glutamine protein, we don't really see anything happening on the bilayer. This was actually just an extra vesicle left behind that gets pounded into the bilayer as we go. And you can see we're in the same spot. If we have now the prolines added to this glutamine stretch, again, nothing happens. If we just add the N17 domain, now we start to see the appearance of all these little oligomers on the bilayer. And if we add the N17 and the polyproline region, it starts to look a lot like exon 1's interaction with lipid bilayers. And so it seems that this N17 domain is needed for this interaction with Huntingtin and the membranes, but it's further modified by the addition of the polyprotein domain as well. And this leads to the obvious question then is there are all of these known post-translational modifications that occur in these 17 amino acids, and how are those going to impact its ability to bind membranes? And these post-translational modifications are linked to all kinds of trafficking signals in the cells and whatnot. And so we've done a lot of work trying to figure out how those post-translational modifications impact aggregation, and specifically how it impacts aggregation on lipid membranes. But I wanted to not focus on that too much to get to some newer stuff. I want to point out that the actual membrane plays a role as well. And so, for example, if you aggregate Huntingtin in the presence of vesicles of just simple lipid systems and track it with thioflavin, you can see some different aggregation patterns. So for example, if you have POPC, if you get to a high lipid to protein ratio, POPC can accelerate fibril formation. On the other hand, POPE actually can start to inhibit fibril formation tracked by thioflavin. These two anionic head groups both have the same impact on Huntingtin aggregation. It increases it, but there's actually this odd thing where as you increase the lipid to protein ratio, the impact goes down. And we think this is because of NC17 has a positive charge. It's highly attracted to this anionic head group. But as you get more lipid, you can actually spread it out. So we think it's a crowding effect in the case of these two things. But obviously the lipid and different lipid components are affecting aggregation. But this is a measure of the whole test tube. So we don't know what's going on the surface or in the bulk solution. And so we wanted to look at some of these things directly with AFM. And here we have brain lipid extract, where we see our normal type of aggregation. And here we added 10% of cholesterol to that. And you can see the morphology changes on those two bilayers are completely different. So lipid composition plays a huge role. And we've looked at a lot of different types of lipid composition. So this is kind of a review of the last slide, the brain lipid extract and the cholesterol. These are actually slides from that actual movie. But we've also looked at things like outer mitochondrial membrane mimics and inner mitochondrial membrane mimics. And those both cause different aggregation patterns directly on the bilayer. So for example, this looks somewhat similar to what we would see with total brain lipid extract on the outer mitochondrial membrane. But the inner mitochondrial membrane, the aggregation looks completely different. And the biggest difference between these two is the amount of cardiolipin in those membranes. We're also recently looking at ER membrane mimics. And here you see something a little bit different. I would like to point out that now we start to see with some regularity these spots that start to turn into fibrils on the surface. And if we just use purified ER and make a bilayer out of the purified ER on our substrate, we get a huge amount of fibrils. The biggest thing though that really causes these fibrils to form on the bilayer appears to be divalent cations. So we get a lot of calcium left over in this. We've run a lot of control experiments. It seems like calcium is playing a huge role via the lipids to enhance aggregation into fibrils of Huntingtin at least. So this led us to the research question that I kind of want to dwell on more specifically, and you can see why it was important from the last talk as well, is what role does the aggregation state play in these Huntingtin membrane interactions? And so what we did is we used this polydiacetylene lipid binding assay. So basically, you take diacetylene monomers and you mix it with your lipid of choice and you make these vesicles. You then expose it to UV light, which polymerizes the diacetylene monomers into a polymer. And the backbone of this, with the size you get basically a nice brilliant blue solution. As proteins bind to these lipid vesicles, they create tension on that backbone, which shifts the color to red. So you get this blue to red color shift that you can track spectroscopically. And so we decided we were going to try to make controlled aggregate solutions and test which ones give us the better color change. So basically here you can see if we just change concentration, for example, we get a much higher signal with more protein as you would expect. So we incubated A-beta for different amounts, or sorry, not A-beta, Huntingtin for different amounts of time, checked the aggregate species by a couple of different methods, AFM being our method of choice here. And so we've got oligomers if we incubate for 1 hour, for 3 hours. After 5 hours, we start to see some fibrils. After 8 hours, we've got a lot more fibrils, and we can basically count the number of aggregates and the type of aggregate in every single image and get enough to get some statistics. And what we see is that the oligomer population goes up to about 3 hours and then starts to decrease, that's about the time we start to see fibrils in these preparations. And so we took these different preps, we staggered the starting time, so at the same time we have all these different preps ready to expose to these PDA vesicles. And this is what we kind of saw. So if we took fresh Huntingtin, we get a signal that comes up to about right here. If we've pre-incubated for 1 hour, it goes up to the green, 3 hours up to the pink. Then after 5 hours, the signal starts to drop. 8 hours, it drops some more. And what we noticed was that pattern kind of mimicked what we saw with the oligomers. So if you actually plot over several experiments, this normalized PDA signal, maximum PDA signal to the number of oligomers in those samples, you get a pretty tight correlation. But of course, none of these samples are really pure. We've got oligomers, we've got monomers, we've got fibrils. So we wanted to try to really control that a little bit better. And so here we've got AFM images of a fresh Huntingtin solution. This was an aggregation of oligomers that we did in the refrigerator to try to slow it down and control it a little bit better so you get a high density of oligomers. And then we also took an incubation that we seeded with preformed fibrils to try to really drive it to fibrillization. And we tested these 3 samples. And again, we saw our fresh sample, our oligomers has a really boosted signal interaction with the membrane, and our fibrils basically kill it off. So we're pretty sure that these oligomers have the highest membrane activity of the different types of aggregate species. So we thought, well, we want to study how this works. But as you saw in the question session last time, there's concern about the transient stability of oligomers. And so we looked in the literature and tried to figure out some ways in which we could stabilize oligomers. Well, like pick up and chick up, we don't have the right amino acids for those types of cross-linking methods. And so we landed on this dinitrofluorobenzene cross-linker that binds to lysine residues. We've got 3 lysine residues in our N17 domain that we know are important for Huntingtin aggregation and actually somewhat for lipid interaction as well. So we thought that was a good candidate to use. We looked at our AFM or our different populations of oligomers. So we decided to focus on the 1 hour time point and the 3 hour time point. So what we did is we incubated for 1 hour or 3 hours, treated it with different doses of this DF-DMB crosslinker. Then we stuck it back in a plate reader to track thioflavin to see if we were actually stabilizing oligomers or not. And we saw something kind of actually odd. So here is after cross-linking after 1 hour. If we didn't cross-link, our control signal continued to goes up with the thioflavin. But with low doses of cross-linker, we actually accelerated fibril formation. But when we get to a high enough dose, we can kill off fibril formation based on thioflavin. If we cross-linked at 3 hours, we saw the same pattern, except now the 5x does not accelerate aggregation. It actually slows it down, right? So here we thought we have a method to maybe stabilize these oligomers. So let's characterize these oligomers a little bit more. And so we incubated our aggregate, or incubated our Huntingtin. And here is what those oligomers would look like before cross-linking after 3 hours. If we take this same sample and don't treat it, after 24 hours, we've got these fibrils and all this going on. If we treat this sample with cross-linker and image it at different time points, so here we've got the 2.5x treatment, the 5x treatment, the 10x on down the line, we can see that the 2.5 treatment results in fibrils, but these fibrils are a little bit different. They're squiggly. They're not as rigid looking as the normal non-cross-linked fibrils, but this correlates well with what we saw in thioflavin. With the 5x, we see some small fibrils appear 24 hours later, but for the most part, we don't have the extent of fibrillization we saw in the control. And then with 10x, 15x, and 20x, this all goes down. And so we did some AFM image analysis of these actual samples here and compared them to this 3 hour time point just to see how stable these oligomers might actually be. And so if you plot the diameter of each individual aggregate versus height, you can see that with the 5x, there's a little bit of change in the overlap of these morphological characteristics, but with 10x, 15x, 20x, they're pretty much right on top of each other. I just want to quickly point out before I flip to the next screen, the diameters we're measuring are right around in that 40 to 60 nanometer range. And that's going to be important for the next slide. So what we then decided to do was to try to figure out how long of a shelf life we had with these oligomers. So we turned to dynamic light scattering to do this. And so 3 days after cross-linking, we subjected these things to a dynamic light scattering assay. So here with our 5x, this is the 3 hour pre-incubation, then cross-linked. The 5x, you can see we get large aggregates. And they look actually larger than the control where we didn't do any cross-linking, but this is a little bit of a misnomer because the Huntingtin was so aggregated that it was off scale of the instrument. So we actually had to do a high-speed spin to pellet some things to actually even just get a reading. And so this is what was left over after that spin. So the Huntingtin actually is more aggregated than the 5X, but the 5X oligomers aren't stable over 72-hour period. But the other higher doses all appeared very small, and we can zoom in on these. So here we can see we did after the 3-day, this is the size of our oligomers with the 10X, 15X, and 20X. And then we did it again 10 days later. So you can see with the 10X, we get a slight shift in the size of oligomers, but with the 15X and 20X, they seem pretty stable. So I feel like we had a pretty good shelf life stabilized oligomers, so we could try to study this lipid oligomer interaction a little bit more closely. And so we went back to our PDA assay, and it turns out that we're not really going to be studying this lipid oligomer interaction the way I thought we were going to be doing it, because it basically cross-linking kills off the interaction. So as we increase the dose of cross-linker, the PDA signal basically disappears, and you can't get any interaction with the lipid bilayers. Well, so we found that a little bit interesting. It wasn't what we were hoping to do, but we decided to work with it. And so we went and we made an oligomer sample, 3 hours of incubation. We made a fibril sample that was seeded so we could get lots of fibrils. And then we had some stabilized oligomers. And we exposed supported bilayers to these different types of aggregates. And so one of the things you see is what we would expect. It's a little bit less aggressive because we had to go to lower doses because of all the cross-linking treatments and dilutions and stuff. But you see development of oligomers and patches of disrupted bilayer regions when we expose these things to oligomers. When we add fibrils, we still see some damage, but we don't see fibrils come down. And this is probably some of the residual oligomers and stuff that might be in there, or stuff coming off the ends of the fibrils, but there is some damage. Cross-linked oligomers, we do see oligomers show up. We don't see any huge amounts of damage on the bilayers. So then we quantified all of this. And so basically the RMS roughness regions, when we have just normal oligomers, get much larger than what we see with fibrils. And if we look at the cross-linked oligomers, the RMS of those surfaces is basically like a clean bilayer. We don't really see any damage. The percent area that's disrupted is completely killed off as well. So we're pretty sure that our cross-linking kills off the interaction with the bilayer, which led us to we tried to figure out why is this happening? What are our hypotheses? Well, we came up with two. One is that we're removing those positive charges associated with lysine, and those might be really important for interaction with the bilayer. We saw this with the acetylation of monomeric Huntingtin earlier in our studies. The other one was that the conformational flexibility of those oligomers might be really important. And by cross-linking it, we tie that up as well. And so we designed this experiment. We wanted to get rid of the possibility of forming fibrils. So we decided to switch to 20Q because one, it forms oligomers, and we knew it would interact with the bilayer as well. The second thing we did is we wanted to disrupt some of the charge associated with the lysines without cross-linking. And so we took a page from Ron Wetzel, and we used these truncated NT17 peptides that we could incorporate into the oligomers. And so we had some that were wild type, and we had some that are had all of the lysines mutated to glutamines to basically mimic acetylation, and those get incorporated into the oligomers. So the first thing we did is just image them to make sure we had oligomers, and then we took those. We could treat all of those with DFDMB as well, and then so we had without DFDMB, with DNB, and we did the PDA assay. So first of all, if you have Huntingtin exon 1 with 20Q, the oligomers give us this yellow signal right here. If we incorporate some truncated NT17 into those oligomers, we get a little bit of a drop in the signal, but it's still pretty strong. It's very comparable to Huntingtin exon 1. If we incorporated acetylated lysine mutations into those NT17 peptides, it drops a little bit again, but it's very comparable. If we cross-link this Huntingtin exon 1 with DFDNB, it completely kills off the membrane interaction, shown down here. If we cross-link the oligomers that have the wild type peptides incorporated into them, so all the lysines are still there for cross-linking, it kills it off. But if we cross-link the oligomers that have peptides with their lysines removed, it drops, but not nearly as much as the other ones. And so what this was telling us is that one, charge is likely still playing a role in this oligomer interactions with lipid bilayer, but the structural flexibility seems to be much more important, because this here, we could effectively remove all of the charges with our cross-linking agent, but we can't make all of the effective cross-links because half of the lysines are missing. And so we think that both play a role, but structural flexibility seems to be a really important aspect of that. So we have these stabilized oligomers and stuff as well now. So we thought, why not see what happens if we throw them at cells? So we took some neuro-2A cells and we treated them with oligomers. We treated them with DFDMB vehicle control, and then oligomers that had been treated with 20x or 5x DFDMB. Unfortunately, while our oligomers were toxic to these cells, so was our DFDMB. And so we couldn't really see anything until we started to dilute the dose. So if we go down to lower doses of our oligomers, they're still toxic. The DFDMB control that gets carried through is still toxic, but we start to see a significant decrease in toxicity associated with the cross-linked oligomers. Then we go much lower, we can basically kill off the toxicity of those oligomers. The DFDMB becomes less toxic, but the Huntingtin oligomers are still toxic. So what we think is going on here is that the DFDMB cross-linking of the oligomers eliminates their toxicity because they can't interact with the membranes as efficiently. But cross-linking with DFDMB, the Huntingtin actually removes the DFDMB from solution as well because it goes into the cross-linking. So we're actually fixing the DFDMB toxicity at the same time. And this is why the 5x is actually better at saving or reducing toxicity than the 20x, because we have much less DFDMB in this sample. So that got us thinking, we can't use DFDMB because it's toxic, but is there something we could do to block these oligomers from binding membranes as a potential therapeutic interaction or therapeutic strategy? So we went back to our simple PDA assay and we set it up to do screens. So here we've got controls with buffer that remain blue. Here we have 3 controls where we added Huntingtin and it turns red. And here we have a positive control where we add NaOH just to make sure our vesicles are working the way they want. And then in each well, we've got a different drug. And you can see after 24 hours, most of them have turned red, except we've got one here that stayed blue. And if we look at the colorimetric response, it basically looks like the control across this row, right? So we screened about 1,500 compounds, came up with 15 leads, retested them, and we have 5 that we think are viable candidates. So for example, if you do a kinetic of these 2 drugs, benzamyl and RO97501, which I'm just going to call RO, you can see them reduce the PDA signal. So here's the PDA signal of Huntingtin interacting lipids with no drug and the addition of the drug. We also validated with a calcium dye leakage assay, and you can see both of these drugs prevent, relative to Huntingtin control, significant amounts of dye leakage from vesicles upon exposure to Huntingtin. And so we wanted to start studying these things in a little bit more detail. So we've done dose-dependent curves. So as you decrease the dose, the PDA signal starts to creep back up. So there's a dose-dependent inhibition of oligomers binding to lipid bilayers for both RO and for benzamyl. Although RO seems to be a little bit more effective, it takes a smaller dose to get a decrease in the PDA signal. So then the obvious question is, how do these things impact aggregation, right? So we looked at the aggregation, and the short answer is, at first glance, it doesn't seem to impact aggregation that much. I mean, you can see we've got oligomers at the early time points. We got fibrils at about the same time point after 5 hours. We got lots of fibrils after 8 hours. So they don't seem to have a huge impact on aggregation, but they do impact the ability of the oligomers to bind the bilayer. So we wanted to get an idea of how these might be working then. So we moved to molecular dynamics. And so we took the structure of a Huntingtin tetramer of the NT17 lipid binding domain that was published by the Chlora Lab a couple of years ago, and we interacted it with the 2 different drugs. With RO, what we found is there's a strong interaction with serine 13, and it appears to fit right into this groove of the tetramer structure of this lipid binding domains of Huntingtin. The benzamyl, there doesn't seem to be a preferential amino acid it's interacting with. We do see a little bit higher preference for 2 of the lysine residues, but it destroys that structure of the tetramer. And so if you look at the MD simulations, these are just for the 4 different chains in the tetramer. the percent of helicity as a function of time, and you can see that this basically gets destroyed by the addition of benzamyl. So this made us think, well, we didn't see anything with aggregation. That doesn't really make sense, but we went back and characterized the oligomers. And what we do see is at the 1 hour, 3 hour, and 5 hour time point is that the oligomers formed in the presence of benzamyl do tend to be smaller than the oligomers of the Huntingtin control or the oligomers that were formed in the presence of RO. So it does seem that even though they're not destroying the oligomer completely, that it is affecting the structure of it. So there might be something there as far as benzamyl really altering the alpha helicity of those NT17 domains of Huntingtin. With RO, we wanted to check that mechanism with serine. We had just published a paper on phosphorylation. So we happened to have a Huntingtin exon 1 construct in which we had mutated that serine to mimic phosphorylation. So we actually add a negative charge there. And so we can do the PDA response control with that. And if we add RO, we can kill off that membrane interaction with a very small dose. So 0.1x, so 1 RO to every 10 Huntingtin, we're able to kill it off. Whereas with the normal wild type Huntingtin, 0.1x is not capable of reducing the interaction at all. So it seems like that serine is important in the RO mechanism of how it's interacting with these oligomers. But we got a lot of work left to do. We're still working on that. So of course we got to do toxicity assays and all that type of jazz. So we're working on that as we speak. But just as a quick, for take-home message, mutant Huntingtin directly interacts with and damages lipids, and it matters on a lot of different things. And even the aqueous solution matters. So if you crowd the aqueous solution, you can change the interaction with the membrane. We recently published a paper on that as well. More recently, we've been focusing on specific aggregate types, and it appears the oligomers seem to be the main culprit in these interactions, which we're going to have to go back and kind of look at some things there as well, because all of our other previous studies, we've worked really hard to start off with monomers as close as we could get. But it does seem that we can directly target Huntingtin to prevent this interaction. And we have some compounds that we think might be viable candidates for this, but we've got to evaluate how they're going to affect toxicity before we can really say anything strong about that. But of course, I'd be remiss if I didn't thank the people who actually are in the lab day in and day out. And so the cross-linking studies were done by Faiza, who recently graduated. My mouse is not working right there. And Chaturanga is the one who did all the drug screens. Of course, there were little bits of pieces from all of my students sprinkled throughout this talk, but those are the two projects I talked the most about. And I also want to mention that Blake Mertz helped us quite a bit with the molecular dynamic simulations that we're starting to do. And collaborator Werner Geldenhuis helped us with all the toxicity assay, or the toxicity assay that I presented today. I'd be more than happy to answer any questions or address any comments that people might have.
Thank you very much, Justin. Great talk. Let's take questions from the Q&A folder. You can also look at them. Let's see here. There are two questions. One is from Soy. Is there a determined site of interaction of the benzamyl with the oligomers? If yes, can you provide some information on the bound structure?
So the only thing we have so far is that MD simulation, and we didn't see any specific specificity, that's redundant. We didn't see any specific residue that it liked to interact with, and it seems to destabilize it. So I don't know for sure if we would have a structure or not. One of the things we were hoping to do is try to do some time-of-flight mass spectrometry studies that might give us some idea of how those things are fitting together. The question is how do they interact with monomers? So we've done some MD simulations with monomers as well, and we get a lot of pi stacking, but again, not along a specific residue. It kind of just stacks all around it. So it's a little bit tricky. It's not like RO where it seems like that serine seems to be really important. We did do a study with mutated lysines to see if the benzamyl had, since there was a little bit of a higher preference for the lysines, and there's a little bit of a change, but the lysines change the interaction of mutating the lysines changes the interaction with the lipid so much that it was really kind of hard to interpret. So we think it might be the lysines, but we don't know for sure at this time.
So in one of your slides, you showed the data with cholesterol. I saw, I think, if I remember correctly, you see a lot of fibers, right?
With the cholesterol, when we have on the bilayer, we actually don't see much fibrillization at all. When we start to see it with the ER. So I thought, let me go back. Yeah, so here you can see up in on the top corner here.
Or the rightmost one is with cholesterol, right?
Yeah, yeah. And so those are like these really weird domains. They look like RAF domains actually, but they're larger, taller above the bilayer than RAF domains. So I don't think
So
they're
domains from the cholesterol-induced RAF domains, not
yeah, that's what it looks like to me. But when you do that,
that makes sense.
Yeah, but like when we have the ER, we get all these fibrils. And these actually we can track them on the surface. They'll grow on the surface. But what we notice is like we've been doing like brain lipid extract forever, and we hardly ever see a fibril. I mean, I could count like maybe every 20 experiments we'll see a fibril. So whereas when we started this, every time we had divalent cations in there, we start seeing fibrils like crazy. So there's something with divalent cations with the membranes that what we work with that seems to enhance fibrillization. And we're trying to figure that out. We haven't figured it out yet. But
it's in your THT data, only in the presence of PE, I think, you don't see, you see inhibition. Inhibition, right?
Yeah,
and,
so it's
no
interesting though. So if we have total brain lipid extract, we see much longer inhibition. And if we spike, and this is something we haven't published yet, we're working on the manuscript. If we spike these systems with cholesterol, they all come down. So for example,
intensity comes down.
They all significantly come down. So like POPG will still be enhanced, but the enhancement's decreased if we've got good cholesterol. If we have POPC, it comes down. There's one of them where it had little effect, and I can't remember which one off the top of my head, where there wasn't as big of an effect. It might have been one of POPS or POPG, but for the most part, most of the systems when we add cholesterol, the fibrillization would go down, measured by thiophoid.
So I wonder, it could be because the physical property of the bilayer, in the PE, you got negative curvature, which is intrinsic. And also in the cholesterol, you have these boundaries between the ordered and disordered domain, which are also curved. So I wonder if that would be destabilized in the fibers.
Yeah, it's interesting. So we also tried, we tried a couple of things. So there was a paper, I forget who published it about 2 years ago, maybe, in which they showed lipids destabilizing fibrils. But it wasn't with Huntingtin, it was another thing. So we actually made fibrils and tried to destabilize them with lipids. And we tried pretty much every lipid we studied, and we couldn't get any destabilization with any of the lipids we've studied. The fibrils were still persistent. On the cholesterol, I don't know. So we have data, we haven't finished analyzing it yet, where we've tried to figure out how Huntingtin partitions to the lipids if it changes with the amount of cholesterol. So we can do these mass spec studies where we get complexation between lipids and Huntingtin and figure out how many lipids per Huntingtin molecule and all this type of stuff, and how many dimers per lipid. And we have cholesterol data for it, but it's a lot of data. We're still combing through it. So maybe we'll see something there where it changes the amount of interactions. But one of the issues we're having is the cholesterol is not showing up in the mass spec very well. So we're having trouble getting the cholesterol into the mass spec. So I wish I had an answer for it, because those domains are really weird looking to me, and I don't know what they are for sure.
I'm curious, in this vesicles THT curve, did you ever see anything beyond 18 hours? Does the intensity stay where it is, or does it come down? Yeah,
so we've gone out to 24 hours is the longest we've done. They tend to stabilize or crash out.
What happens if we go beyond that? Do you see fiber remodeling?
Not really. So the better set of data I would have for that would be when we made fibrils and then added the lipids, and we don't see any change that way, right? So
what about for A beta? Did you see anything for A beta, anything like that?
We haven't tried it with A beta. We've been focusing on Huntingtin with that. So yeah, the paper that I think I saw was an alpha synuclein paper. I could be wrong, but we tried that and we didn't see anything with Huntingtin. I mean, we do like 90% Huntingtin now. We haven't been doing as much A beta recently. So I have not seen it, but Huntington's a bit weirder
Yeah.
than some of those other proteins because of that homopolymer stretch, right? You don't get as much polymorphism and stuff like that because it's kind of self-correcting. If you see some like Patrick van der Waals NMR papers and stuff like that,
Right.
that the glutamine's kind of, there's only so much you can do with a polyglutamine as far as stacking them into a fibril. So they might be a little bit different than some of the other ones.
So there are several questions in the Q&A box. If you can see from the top, let's go from the top. The first one is from Murugesan Velayudham. Does HTT aggregates interact with the nuclear membrane? What about the interaction with the HTT aggregates and cellular proteasome?
Yeah, so with the nuclear membrane, that one I know, and it's something that I've always wanted to kind of look at, but we haven't gotten to it yet. So one of the features, and so one of the classic Huntington mouse models is R62. And if you look back at some of the early papers, there's actually this really weird increase in nuclear pore complexes in the R62 mouse, which to me suggests that Huntington is impacting the nuclear envelope somehow. I've not actually investigated it myself yet, and I haven't seen anybody follow up on that observation. But my guess is it probably will interact strongly with the nuclear envelope. With cellular proteasome, yes, it does. So there was a paper by David Smith a couple of years ago. He's also at West Virginia University, and it focused mainly on A beta because they could stabilize the A beta oligomers better. But that seemed like they were getting interaction of Huntingtin. We gave them our Huntingtin constructs with the proteasome as well directly. So it does. Again, I've not studied that myself directly, so I can't give you much details, but I can say that it definitely does seem to directly interact with the proteasome as well.
There's a question from Charlie Brooks in the chat box. Are all of the lipids, lipid concentration above the CMC? I think they are, right? You're using liposomes.
Yeah, we're above the critical micelle concentration. So
what happens if you do below CMC? Do you see any?
So we tried it. So we actually just got a reviewer for one of our papers mentioned this, and so we've tried some things, and so we haven't repeated everything yet. But here's what we kind of see. So one thing, if we try to do that with mass spec, we don't see complexation. We can see the free lipid and we can see the free Huntingtin. Whereas if we do them with vesicles, we see complexation where we can see Huntingtin grouped with different lipid species. The other, and that actually has been published a couple years ago with Dr. Valentine, one of my collaborators. The other, we recently tried it with our PDA assay. So we first tested to see if we added a little bit of extra lipid after we made stable vesicles, if we could see a change in the signal, and we weren't inducing any change in our signal just from adding lipid. So then we mixed lipids and a low dose of Huntingtin to see if we could see any difference in how much Huntingtin would bind to those vesicles then with free lipid available. And we've done it once. We actually did it last week for the first time, and we didn't see a change, but we haven't optimized. There might be some optimization that needs to be done. So as of right now, I don't have any evidence for it. I don't have any evidence against it either. And so I guess long story short, I don't know. It would be, I guess, the way to put it.
Let's look at the questions in the Q&A folder. One from Cesar Avila. Thanks for such a great talk. I might have missed it. How do you separate or purify oligomers from monomers and fibrils?
Yeah, so that's the tricky part. So in that first set of data I showed, we couldn't do it, right? We just incubated for a certain amount of time. We checked as best we could through AFM what the actual population looked like, but we couldn't separate it. And so that's why we did the second experiment where we took Huntingtin that we high speed spin, fresh, and we didn't even let it sit around at all. That came out of dialysis from purification. We interacted it with the bilayer, and then we tried to slow down the oligomer formation as best we could by doing it in the refrigerator for a certain amount of time to try to get an oligomer population without Huntingtin. And then the other one, to get fibrils, we just seeded to try to get as much fibrils as possible, and then we could see a bigger difference. But we wanted really to have one of those nice pure samples, right? Because we know a little dose of something sometimes can make a big difference. That's why we went to the cross-linking, but it kills off the interaction. right? So it led us in a different direction, but that's what we were aiming to do. That's a real tricky thing to get around, is how do you get a pure sample of some of these aggregates? And even with oligomers, you can get pure oligomers, but you can get such a size difference in that sample, right? And from the previous talk, you can see the importance between low molecular weight and high molecular oligomers in A beta. So how do we control for all the heterogeneity in those samples? It's really tricky. And we have not figured it out completely. We try the best we can.
So the next question is from Sai. Are the interactions between the membrane and the MHTT random as the aggregates float around in cytoplasm? Or are these interactions occur as a consequence of association between MHTT and cytoskeletal components, such as microtubules, motor proteins, etc.?
I would want to say I don't know for sure. We haven't done much work within cells with all those different components. We've done mainly with purified systems. I wouldn't say that the interaction is random in the sense that there's clearly being trafficked, right? So for example, Ray Truant showed that oxidation can change the location of Huntingtin from the ER membrane into the nucleus, and there's other nuclear transport signals and stuff like that. Simulation changes the location of Huntingtin through that lipid binding domain. So it's definitely being controlled somehow. How it's being controlled, I don't know for sure. Well, at least I can't say from my studies because we haven't done much cell work. I would say one thing, this is a little bit unrelated, but we published a paper a few years ago with beta amyloid, in which we disrupted cytoskeletal components. And we could see a difference in how cells responded to A beta binding to the plasma membrane by different states of the cytoskeletal elements. And basically, if you, how a cell would respond based on that to exposure to A beta was different. And we can measure the mechanics of those cells and everything like that. So at least with A beta, I think that's I can see where that question would come from, right? But I don't know for sure with Huntingtin.
Great. There's the last question in the Q&A folder from anonymous attendee. This is an extension to the question, I think, from me. Did you study how different vesicle diameters affect the fibrillization?
Yeah, so the answer is no, but we tried to. So we found that with our lipid preps that we were getting with those same types of, with those different lipid systems, we were getting roughly vesicles with the same diameter, right? And so we felt that was, we actually for that study wanted them to be the same diameter to try to avoid an issue with that. We had trouble getting a range of diameters for that as well. A long time ago, we actually tried to look at curvature through direct observation with AFM. And so what we did is, so basically there's this really cool paper that came out where they put beads on a substrate and then they grew the bilayer over the substrate. And they had different sized beads and everything. So we thought, well, if we just put beads of different size on our substrate and then put the bilayer on top, we can do the AFM on the precisely controlled curvatures. The problem was with the lipids we were using, we could only get them over one size of beads. All the other beads would just grow around them and leave like a hole, you know? And so we did do the study with curvature with that one size bead. And it does make a difference. Huntingtin has a high affinity for curved membranes, at least in vitro, what we've seen. And this makes sense when you look at, so there's a lot of antipathic alpha helix that go from intrinsically disordered to lipid binding that have a high propensity to bind curved membranes. And that's exactly what the anti-17 structure kind of behaves like. So it's not surprising. Curvature is definitely probably going to play a role in those interactions, and that's going to then play a role downstream to fibrillization and all of that. It's just we've had trouble controlling those diameters well enough to really answer it. But I think that's exactly what's going to happen. That would be my guess.
Well, thank you very much, Justin. We answered all the questions. Let's bring in anyone who wants to join the panel. Please raise your hand. We can promote you to the panel. We can take questions for both the speakers. Kenjiro-san, are you awake? Yeah, one person saw he raised his hand. So Kenjiro-san, I have a question for you.
Yes.
do your oligomers, do you try to grow the oligomers in the presence of lipids? And did you end up with the same type of oligomers? If you did, or did the oligomers what you got when you try to grow in the presence of lipids, do they vary depending on the lipids?
Yes. This is also important. Yes, my interest is also same lipid change, structure, and biological property of oligomer. But and so as you suggested in my presentation, I have to next stage. I have to check. This is important. Thank you so much.
Sai, you are promoted to the panel. You can ask the question directly. Go ahead, Sai. You would unmute.
First of all, thank you so much, Professor Justin. It was so interesting to listen to your talk, and Professor Kenjiro, it was interesting to listen to your talk as well. So my question is that since there were promising results with respect to benzamyl and the oligomers, do you plan to assess its metabolic toxicity? Like, do you consider checking at its metabolic toxicity before going to further analysis?
Yeah, well, benzamyl is an approved drug for treating acidosis already. So, and actually what's kind of interesting is there was a paper already published where they tested benzamyl, wow, it was about 10, 12 years ago, in the R62 mouse of Huntingtin, and actually showed an improvement in Huntingtin, but they were checking just to see if you altered, acid sensing ion channels or not, which is what benzamyl does for its normal thing. So it's already got some approved use as a drug. So I don't think I would necessarily need to go too far into it. But interesting is it did improve the HD phenotype in the R62 mouse.
Yeah, that's what.
Say what?
Yeah, go on.
So we're just seeing that it interacts directly with Huntingtin as well, because in our screen assay, all we have is lipid and all we have is Huntingtin. And if it interacted with the lipid, it would have caused the color to change, right? And so we know it's interacting with Huntingtin, so there might be more to the mechanism of action where how they saw that improvement in phenotype in that R62 study that was published a couple of years ago.
Thank you.
Velayudham, you can unmute, ask a question.
Hello, Justin. It's a great talk.
Thank you.
I have one general question, okay? It's like, for example, okay, in COVID-19 patients, they have increased some kind of neurological problem, So in that case, what about the COVID-19 infection, whether it can increase the Huntingtin aggregation?
I don't know for sure how that might impact it. So with Huntingtin, would be kind of interesting because most people don't have a mutated Huntingtin protein, right? But in vitro, you can get non-mutated Huntingtin to aggregate. So it'd be kind of interesting question to see if there is, I don't know how you would go about it, but what could cause you to invoke Huntingtin aggregation in a non-mutated form to maybe look at some of the initiation processes. I don't know of anyone who's looked at COVID-19 specifically. I know we can seed it, like we can take Huntingtin seeds from mutant Huntingtin and seed non-mutant Huntingtin to get aggregates. So yeah, it's kind of an interesting thing, but that goes with almost any protein that forms amyloid. It's like what environmental factors, or like why does one person get Alzheimer's and not the next when they don't have any of the hallmark or any of the familial forms or any of the other risk factors? Like what causes that? I think there's real subtle differences that can tip the balance of protein homeostasis that could be interesting, and COVID-19 infection obviously could be one of them. But I don't know any direct data about that. It's an interesting question though.
Yeah. I have another simple question, okay? So when you talk about the mitochondrial outer membrane, there are some proteins on the outer membrane of the mitochondria, okay? Is there any interaction between the Huntingtin as well as the other proteins on the mitochondrial outer membranes?
Yeah, so there's a group that's been looking at that specifically. I forget there's one protein that the oligomers interact with specifically. In fact, it's kind of interesting because a lot of the cellular studies, if you look at them, Huntingtin oligomers tend to accumulate around the mitochondria, whereas there's a really cool Cell paper where they show fibrils around the ER. So it's kind of an interesting thing there. But yeah, they do. We couldn't pick that up in our studies. We did in our study. I didn't show it, but we've published it. We did purify out intact mitochondria to try to get the direct aggregation on that with all the proteins and everything intact. We had some technical issues, but in short, we could kind of see that in the presence of mitochondrial surfaces, the fibrillization rate decreases. But that can be lipids, that can be proteins, so we couldn't really pull out the exact culprit in the way we were going about it. But there is direct interaction between mitochondrial proteins and Huntingtin. Other groups have shown that.
But what about the interaction between the Huntingtin and the cardiolipine? Is there any direct interaction between them?
Yeah, so in our paper, the biggest, so we could mimic all the aggregation effects we were seeing with mitochondria in vesicles by altering cardiolipin composition. So if we put more or less cardiolipin into our vesicles, we could get the signals to shift like they did with the mitochondria. So I think the cardiolipin is and a little tiny bit of it makes a big difference as well. So there's definitely a modifying effect of cardiolipin in the membrane on Huntingtin aggregation at that membrane.
Thank you. Thank you very much.
Thank you very much to Justin and Kenjiro and all the participants. Lovely talks, great discussion. I think there are no more questions from the panel, from the participants. So you did a great job. Thank you very much for joining us.
Thank you for the invitation.
Thank you so much for this invitation.
So let's close the session and see you. Bye-bye.
See you.
Bye.
Thank you so much. Thank you.
Now you can go back to bed, Kenjiro-san.
Thank you. Thank you. ありがとうございます。
ありがとうございました。